Hello guys, welcome back to my YouTube channel that goes by the name Life Sciences by Tumi Silani. We are just back from the previous lesson, which was based on the history of the discovery of DNA. If you enjoy content to my video, please remember to like, contribute to the lessons by commenting, subscribe to the channel, and share the link to this video to your fellow learners, teachers, and anyone else who might benefit from these videos. My name is Tumi Silani. I am a teacher at Milner High School here in the Northwest Province. On this lesson will be lesson 5 on the lesson series, The DNA Code of Life. And on this lesson, we will be looking at the RNA structure. On the previous lessons, we've been talking about DNA, what DNA is, where it is found, how it functions, the significance of DNA. So if you're interested to know about that, this series is a lesson build up. So please go back to those previous videos and to get context on the following videos. Like I said, this is a lesson series. I've decided to divide the lesson series into 5 lessons. Each lesson has its own video. So let us look at the breakdown of the lesson series. Alright guys, so the lessons are broken down into 7 lessons, each comprising of its own video. So far, we have dealt with the first 4 lessons. On today's lesson, we will be looking at the RNA structure. The following lesson, which will be lesson 6, will be on protein synthesis. The lesson will be divided into 2 parts. The first part will be protein synthesis, which comprises of transcription, movement of mRNA, as well as translation. The second part of the lesson will be on amino acid coding. The last lesson, which is lesson 7, will be on the topic, or rather the subtopic, DNA profiling, where we will be looking at the definition and uses of DNA profiling, factors that contribute towards the reliability of DNA profiling, and we will be doing some interpretive work where we are analyzing DNA profiles in order to determine paternity, and we will be looking at how DNA profiling is used as biological evidence into determining a possible criminal. So, without any further ado, let's go get right into today's lesson. Okay, remember that the cell contains 2 types of nucleic acids. The first one we dealt with quite extensively in the previous lessons, which is DNA. That stands for deoxyribonucleic acid. The subject of today's lesson is the second nucleic acid, that is known as ribonucleic acid, that is abbreviated RNA. On the following slides, we will be looking at the structure of this second nucleic acid, which is called ribonucleic acid. Okay guys, now in the following couple of slides, we will be looking at how RNA is structured, or how RNA is made. Okay? Human cells contain 2 nucleotides, one being deoxyribonucleic acid, abbreviated DNA, and the other is ribonucleic acid, abbreviated RNA. Now, if you're asked if you get a question referring to a nucleic acid, you must know that we have two, DNA as well as RNA. We have quite extensively looked at DNA in the previous lessons. So here we will be looking at DNA, and the way that I'm going to do it, with each bullet or each description that I will be giving, I will be comparing RNA to DNA. So go back to the lessons, make sure that you are fully versed with the content of DNA, because I'm going to be doing a direct comparison between elements of RNA versus the elements of DNA as I go along. RNA is relatively shorter compared to DNA. Remember, we said DNA is a long molecule, okay? It's about 5 meters long when you stretch it, and when you uncoil it, some sources say it's about 7 meters long. And compared to DNA, RNA is shorter. Hence, I say relatively shorter. RNA consists of 1 strand instead of 2. Remember, we said that DNA consists of 2 strands, but when we look at RNA, RNA only consists of 1 strand. Another point, because DNA has 2 strands, those 2 strands are joined together by weak hydrogen bonds. However, since RNA consists of only 1 strand, there is no bonding of 2 strands, okay? It only consists of 1 strand, therefore it is void of weak hydrogen bonds. It has no weak hydrogen bonds. RNA is also made up of monomers. Remember, the word monomer means building block. Remember, like we said, large molecules within the cell do not come as complete, as large as they are. They need to be built up to be large. Now, the monomers or building blocks of RNA and DNA, quite frankly, are called nucleotides. Both DNA and RNA contain nucleotides, but there are specific differences between DNA nucleotides as compared to RNA nucleotides. So let us firstly look at the general structure of a nucleotide, and we will look into those specific elements that distinguish a DNA nucleotide from an RNA nucleotide. Okay, just like DNA, one nucleotide is made up of 3 components. The word component means parts, okay? So, an RNA nucleotide also contains 3 components or 3 parts. The first one being a phosphate group. Now, I'm going to caution you again, like I did when we were discussing the DNA nucleotide. Please do not say phosphate. Please refer to this as a phosphate group. So the first component is a phosphate group, not just phosphate. I know that a lot of publications, such as textbooks and study guides, say phosphate, but please say phosphate group, or it is sufficient to say phosphate portion. It is not made up of 1 atom of a phosphate, it's actually a group of phosphates. So the first component or the first part of an RNA nucleotide, just like in DNA, is a phosphate group, just like we see over there. The second component will be a sugar portion. There we go. And then the last one will be a nitrogenous base. So this whole thing is one building block of RNA. Okay, guys? So this whole thing is a monomer of an RNA. It is a nucleotide of RNA. It consists of 3 parts or 3 components: a phosphate group, a sugar portion, and a nitrogenous base. Another cautionary warning, please do not use the noun nitrogen base, even though your textbook may say that it is a nitrogen base. please use the adjective nitrogenous base. So it is a base that is nitrogenous, not nitrogen base. So, in the portion that is for the nitrogenous base, an RNA nucleotide may contain the nitrogenous base adenine, uracil, cytosine, or guanine. Okay? Even though we have 4 different nitrogenous bases, one nucleotide only contains one nitrogenous base. So each nucleotide may only contain one nitrogenous base. That nitrogenous base may either be adenine or uracil or cytosine or guanine. It may not contain all 4 nitrogenous bases. Okay? One nucleotide may only consist of one nitrogenous base. Now, the structure that we have over here, this is a diagram showing the general structure of a nucleotide. All right. An RNA nucleotide contains ribose sugar. Now, remember, we have 2 nucleic acids within the cell, DNA or RNA. Okay, guys? Now, both of them are made up of nucleotides, but there are key distinguishing factors that differentiate between a DNA nucleotide and an RNA nucleotide, and you must know those key differences. So, on the sugar portion of the RNA nucleotide, the sugar portion is made up of ribose sugar. However, in a DNA nucleotide, we will instead have deoxyribose sugar. The other thing that differs in a RNA nucleotide is the fact that uracil is a nitrogenous base that is exclusively found on RNA and RNA only. Remember, the opposite of this is thymine. Thymine is found on a DNA nucleotide and a DNA nucleotide only. So instead of thymine, the job of thymine is replaced by uracil. So whenever you are looking at a strand of a nitrogen, or a strand of, whenever you're looking at a strand of a nucleic acid and it contains uracil, you must know that that strand or that nucleotide belongs to RNA. However, if you see it containing thymine, you must know that that strand or that particular nucleic acid is that of DNA. All right, now we said that we have uracil in an RNA nucleotide. So uracil is complementary to adenine, meaning it replaces thymine. It does not bond with thymine. So if on one strand you have adenine, in DNA it will bond to or be complementary to thymine. However, in RNA it will bond to or is complementary to uracil. While in the making of DNA strand, A bonds with T, in the making of the RNA strand, A bonds with U. This is very important. So there we have a diagram showing the structure of an RNA nucleotide. We would have a phosphate group. We would also have a sugar portion, but remember, because this is specifically an RNA nucleotide, we need to specify that in the sugar portion, the type of sugar is ribose sugar. And then the nitrogenous base that is exclusively found in RNA is uracil. Okay, so the key difference between a DNA nucleotide and an RNA nucleotide is on the sugar portion, RNA would contain ribose sugar, but on DNA it will consist of deoxyribonucleic acid. No, excuse me. On an RNA nucleotide, the ribose sugar is on the sugar portion, and on a DNA nucleotide, we have deoxyribose sugar. Pardon me for that mistake. And there is a nitrogenous base that is only found on RNA, and that nitrogenous base is called uracil. However, the nitrogenous base that may be only found on DNA would be thymine. Okay guys, we are continuing with the RNA structure. The number of adenine bases is approximately equal to the number of uracil bases, and the number of cytosine bases is approximately equal to the number of guanine bases. Now, remember this. Cytosine, guanine, and adenine are found on both DNA and on RNA. Okay. However, uracil is only found in RNA, and thymine is only found in DNA. All right. Now, if we have 50,000, for example, adenines, we approximately will have 50,000 uracils. Cytosines, if we have 10,000, we will approximately have 10,000 guanines as well. So the number of cytosine and the number of guanine are the same. Same goes with the number of adenine, and the number of uracil bases as well are the same. So we therefore say that adenine is complementary to uracil and cytosine is complementary to guanine. This is in RNA. During formation of an RNA strand, nitrogenous bases bond in the following way. If on one strand you have adenine, on the other strand you will have uracil. If on one strand you have cytosine, on the other strand you will have guanine. Okay, guys? So this is complementary base pairing in RNA. So remember the analogy that we used when we were describing complementary base pairing in DNA. We said that the apple falls from the tree. So in DNA, A always goes with T. The car parks in the garage, therefore C always goes with G. Okay, but remember, this is RNA. This is not DNA, this is RNA. So what analogy can we use? The car still parks in the garage. So guanine is also complementary to cytosine here in RNA. So this is good and well. Okay, so this complementary base pairing still remains. However, now check. Adenine in DNA is complementary to thymine. However, in making RNA, adenine is complementary to uracil. So, adenine is like a polygamous man. Okay, in DNA, he takes the wife thymine, but in RNA, he takes the wife uracil. Okay, I hope this makes sense to you as much as it is making sense to me. Sometimes it surprises me the things that I think about. So in DNA, adenine will bond with thymine, but in RNA, adenine will bond with uracil. So let's use this analogy rather. The apple sits under the tree. Therefore, in RNA rather, we use U, The apple sits under, this is in RNA, and then in DNA, we use T, the tree. So in RNA, the apple sits under U. In DNA, the apple sits under the tree, T. Okay, I hope this is making sense to you as much as it is making sense to me. If it is not, just memorize it as it is. Okay, we are still looking at the RNA structure. Just to give you a visual there, DNA, like we said, is long and it is a double helix, meaning it has 2 strands and it is twisted. Okay, however, as you can see over there, RNA only consists of only 1 strand instead of 2, such as in DNA. An RNA nucleotide has 3 components: a phosphate group, a ribose sugar portion, and it may contain the nitrogenous base uracil. However, a DNA nucleotide also contains 3 components. A phosphate group, however, in its sugar portion, it would consist of deoxyribose sugar, and it may contain thymine instead of uracil. Okay, the first of our activities, you are to tabulate 4 structural differences between DNA and RNA. 4 structural, guys, differences between DNA and RNA. Not where they are found, not anything else, but structural differences. So this is differences in its structure. Okay, guys? So structural differences. And the question also says tabulate. The word tabulate means put in a table. So let us check. So there is our table. Now, let's check the differences. So you draw your table. On one side you include DNA, and then on the other side you will include RNA. DNA is a relatively long molecule for 1 mark, and then RNA is relatively short molecule for 1 mark. RNA consists of 2 strands, 1 mark, while DNA consists of 2 strands, 1 mark, and then RNA consists of only 1 strand, another mark. DNA contains deoxyribose sugar, 1 mark, while RNA consists of ribose sugar, 1 mark. The last difference, DNA consists of thymine as a nitrogenous base, 1 mark, and RNA consists of uracil as a nitrogenous base, 1 mark. There's a few things that I'd like to caution in such a question. Whenever they are asking you to tabulate, you must put in a table. And then you will see, usually the marker will write the letter T and will either make a tick or make a cross next to the T. This T is for following the instruction. The instruction says tabulate. Okay, so if you put it in a table, you get 1 free mark. But a lot of people are of the misconception that this T is for drawing the table. This T is actually for the relevant headings on each column. If the instruction says tabulate 4 differences between DNA and RNA, so these headings, DNA you put there and RNA you put there. If you put anything else besides DNA and RNA, let's say for example you say DNA nucleotide and you say RNA nucleotide, you will be discredited this mark of the T. So please make sure that the headings of your columns are relevant, and then that relevance you get from the description of the statement that you are given by the examiner. Another thing that I would like to caution, please make sure that when you give differences, you do not compare apples to oranges, but you compare apples to apples. Compare directly, please. Compare directly. For example, when you are comparing the length of DNA, you must correspond it with the length of RNA. You may not say DNA is relatively a long molecule, and on this side you say it consists of one strand. That does not make sense. If you're saying DNA is a long molecule, compare that to the length of RNA, not how many strands it has. So when you say DNA is a long molecule, say that the RNA is a short molecule. Okay? Compare apples to apples, not apples to oranges. Another thing, you must compare directly and not indirectly. For example, we see at the marking center, learners might write, for example, DNA consists of 2 strands. And then here, instead of saying RNA consists of 1 strand, a learner might write RNA does not consist of 2 strands. If you're telling us how many strands DNA has, tell us how many strands RNA has as well. Okay, guys? So do not say DNA consists of 2 strands and then here say RNA does not consist of 2 strands. You must say RNA consists of 1 strand. Same goes here. When you say DNA consists of deoxyribose sugar, do not say RNA does not consist of deoxyribose sugar. Rather say DNA consists of ribose sugar. Don't tell us what it does not have. Tell us what it does have. So, moving on. The marking guideline, let me just erase there. Excuse me, let me just erase there, as well as there. Okay, the marking guideline for this table, you have any 4 corresponding differences. As you can see, we have 4 corresponding differences here, and it is times 2. So it's 2, 4, 6, 8. Okay, so that is the 8 marks. And then, like I said, you get 1 mark for the table. So like I said, you will see the examiner writing T there and making the marker rather, writing T there and making either a tick or making a cross. That is for the relevant headings of the table itself. Okay, caution, mind the following questions. Tabulate the differences between the structure of DNA and the structure of RNA. This is not the same question as tabulate the difference between a DNA nucleotide and an RNA nucleotide. There's a few learners every single year at the marking center that gets these 2 questions confused. When they say give the difference between a DNA structure and an RNA structure, you may give any differences in terms of their structure. However, the second question is asking give the differences between the nucleotide of a DNA and the nucleotide of an RNA. So for example, you cannot say that DNA is a long molecule while RNA is a short molecule. You'll get this wrong. The focus of this is on the nucleotide only as opposed to the structure of DNA and the structure of RNA. Here you can give any structural differences, including those of the nucleotides. But this one is more specific. The differences that you must give are the differences on the nucleotides and the nucleotides only. Versus, okay, So remember to read the question over and over again to understand what it means, or else you are going to lose marks. Let us now move on to the second activity. All right. Study the diagram below and answer the questions that follows. Okay, guys. Now, let us study the diagram. Remember, when you study the diagram, if there are any loose ends, you need to label them. You need to write them down. Okay, guys? Let us now study the diagram first before we look at the questions. So here is a diagram showing molecule 1 and molecule 2. So what this should say to you is that these can either be different molecules or they can be the same molecule. Now, let us check. This molecule over there contains the nitrogenous base uracil, contains the nitrogenous base U. So it means that this strand is a strand of a molecule being RNA. Okay? And then when you look at molecule 1, it contains the nitrogenous base T for thymine. Therefore, we know that thymine is only found in the molecule which is DNA. So I submit that molecule 1 is DNA. Why? Because it contains thymine as a nitrogenous base, and molecule 2 is that belonging to RNA. Why? Because it consists of the nitrogenous base U. Another thing, there are no weak hydrogen bonds. Therefore, these two diagrams do not belong to the same molecule. In face value, you might think that this is DNA because it has two strands, but in further analysis, you see there is uracil. So this cannot be a DNA molecule. This must be RNA. And that strand over there, because it consists of thymine, must be a strand belonging to a molecule which is DNA. Now, what is the question? Question 1.1, identify molecule 1 and 2. On top of that, provide one visible reason for each answer. So it is going to be 2 for the names of the molecules, and then 2 to the reasons why you say each molecule belongs to which which nucleic acid. So molecule 1 is DNA because it consists of thymine as a nitrogenous base, and molecule 2 is RNA because it consists of uracil as a nitrogenous base. So that's where we get our 4 marks. So that is the second and last activity for this lesson. We have come to the end of lesson 5, you guys, which was based on the RNA structure. I hope that this content was very informative for you. If it was, please do like. If you have any contribution, please let me know in the comments. I will read each and every single comment, and I will try to respond to every comment. Subscribe to the channel and share the link to this channel to your fellow learners and friends, so that to maximize their learning potential as well. Thank you so much for watching. The next lesson will be lesson 7. Nope, the next lesson will be lesson 6. That is the protein synthesis and amino acid coding. Thank you so very much for watching, and as usual, I appreciate you. See you next time. Bye-bye.